Hello everyone, this is Mr. Vijay Vik, Assistant Professor, Department of Microbiology, Sophia College for Women, Empowered Autonomous. This is a course on monitoring and control of parameters in an industrial fermentation. An industrial fermentation is a biotechnological process where microorganisms are grown on a large scale in a device called as fermenter for the commercial production of valuable products. The first lecture of this course will be Introduction to a Microbial Fermentation Process and to a Fermenter, where we will learn the basic design of a fermenter and its parts and their significance. The second lecture, on which the entire course is based upon, monitoring and control of parameters and sensors used to monitor those parameters. The next lecture will be monitoring and control of temperature. Microorganisms have an optimum temperature for growth. So let's say if a fermentation process goes on for 7 days, during the 7-day period, the temperature has to be maintained constant. And if there's a deviation, it has to be corrected because it will affect the fermentation process. Monitoring and control of pH is as important as temperature because the pH may not remain the same after inoculation of the broth. And yes, buffers are added to the medium. Carbon and nitrogen sources are controlled, but there is sometimes need to add acid or alkali during the fermentation to the broth. Inoculum, fermentation medium, acid, alkali, antifoam, all these are added to the fermenter via pipelines, and air also enters via sparger but through the pipeline, sterile air. So flow rate, monitoring and control of flow rate of gases and liquids becomes highly significant. So is the pressure, which we'll see in that particular lecture. Foaming occurs during a fermentation and it has to be controlled because it will massively affect the fermentation process, sometimes resulting in a loss of a fermentation process. So antifoams have to be added. Monitoring and control of foam is highly significant. Microorganisms will not grow unless and until they get dissolved oxygen. So monitoring and control of dissolved oxygen is very important. Monitoring and control of inlet and exit gas composition is also something that we will learn. This course is an undergraduate level course, specially designed for TYBSc students who are in either semester 5 or semester 6. Postgraduate students can also attend this course, and even students who are aware of basic industrial microbiology or a fermentation process are also eligible to attend this course. It's an independent course and no certificates will be provided. It's only for learning. Apart from the recorded lectures or videos, study material will be provided. Assignment and MCQ quiz test will be posted in the chat section. Learners can ask their queries in the YouTube chat section. And I believe you will enjoy this course and enter into the world of fermentation technology. I wish you a happy journey.